Hey y'all, Conlon Lab here with the Straight From a Scientist podcast. This is episode 53. It's with Jenna Beam from the Conlon Lab. First off, this is our first episode in 2020, so Happy New Year to all. And I think you'll enjoy what we have coming in the new year. Some housekeeping stuff. Of course, this is a video episode. If you're just listening, you can head on over to YouTube and see the live video interview we did with Jenna. And yeah, it's a lot of fun. She talks about a lot of things about Staphylococcus aureus, which is a bacteria that lives on your skin and can become pathogenic, and of course, MRSA, flesh-eating bacteria, all that good stuff. So as you are executing on your New Year's resolutions, going to the gym a little bit more, do try and be conscious of it, and just take a shower afterwards. Make sure you stay clean, and you leave spaces clean for other people. Just be considerate, right? But we go into the science of Staph aureus, and then also her lab just recently published a paper talking about how Staph aureus might be hiding out in the very immune cells that are tasked with getting rid of it. So that's kind of scary, but really, really interesting, of course. Thanks to Jenna for coming on. And before I let you go, just some housekeeping stuff. Again, this is our first video episode, episode 53. Sorry, our third video episode. This is episode 53. So we have two behind this one on YouTube. If you want to see a live kind of feed of scientists in their natural habitat, this is a great way to do so. You head on over to YouTube, and of course, all the episodes will be available on all the normal audio formats. We thank you all for listening over the past couple of years. It's been great. We have many more great things to come. If you like the podcast, please go us and review us on iTunes, or subscribe to the YouTube channel. There's a bunch of different ways. You can find us on Spotify as well, Stitcher, and if there's any platform that you want us to upload to, you can let me know as well. I'm looking to check all the boxes in 2020. So enjoy, all. Conlon Lab here for another Straight From a Scientist episode. I have with me Jenna Beam, who's a PhD candidate here at UNC in micro and immunology. Are those two departments?
No, it's the same department.
Okay, very cool. So yeah, in the microbiology and immunology department here at UNC, and she studies, in a nutshell, and we'll get into it, bacterial tolerance / resistance.
Focus on tolerance, yeah, but resistance always comes into play.
Particularly in one strain of bacteria that's really known for infecting humans. Rather pathogenic. So it's called Staph aureus. But Jenna, if you want to just take it away and let us know, like, what and why are you working on?
Okay, so I guess Brian's big thing. Brian is my PI, Brian Conlon. He comes from Kim Lewis's lab at Northeastern in Boston, and they are sort of tackling this big question of why antibiotics don't work in staph. So I think it's like in the United States last year, like 120,000 people or something had a staph infection, and 20,000 people died. And we have all these antibiotics, and they don't work. And nobody really knows why. It doesn't seem to be entirely resistance. Like, there's definitely some of that comes into play. But there seems to be some other facet of staph's lifestyle when it's infecting that stops the antibiotics from working or prevents them from working. And so when Brian started his lab here, he was really interested in probing these antibiotic tolerant cells. They're sometimes called persisters. That definition is kind of ambiguous in the field. But specifically, my part of the lab, my project, looks at how staph interacts with immune cells and how this interaction can affect tolerance during infection.
Very cool. First off, thanks for coming on. I don't know where my manners went.
Thanks for having me.
I really appreciate it. It's great to have people like tell their stories and their science. So I'm gonna back up and try and catch myself up too, because there's a lot of field specific lingo there. Yeah. So Staph aureus, again, it's probably best known for skin infections.
Yeah. So skin and soft tissue infections are like the sort of normal staph infection, right? The really nasty, like pussy skin infections.
I think of it in the context of like gym or like wrestling mats.
Yeah, so I always talk about that. Yeah, I always talk about like it's the kind that athletes tend to get. Yeah. And I realized that like if you haven't been in a locker room or been around athletes, you probably haven't seen that. So I've stopped using that as like a point of reference. But yeah, that's exactly it.
Yeah, I mean, I know it myself. I was doing jiu-jitsu for a while. I didn't get Staph aureus. I got a different infection. So,
Great.
Yeah, I mean, it is close to home for me, though, because I know many people have. But so you said 120,000 a year, but 20,000 die. So 1 out of every 6 people can die from that. I didn't realize it was that bad. I thought it was just, you get it, you get an antibiotic cream, and then you go home and you're fine.
Well, yeah, so like in a normal, healthy high school wrestler or whatever, right? Like they have a perfectly intact immune system. And so it gets treated. They notice they have some sort of infection. They put an antibiotic cream on it and it goes away. But it's actually really common in IV drug users who use dirty needles. They get these sort of like deep-seated infections and they don't seek medical attention. And so when they sort of go septic is what happens. But when they don't treat these infections, staph can get into the bloodstream and get really nasty. It's common in like ICU patients with burn wounds and stuff like that.
So anytime it has like a clear access point into the bloodstream. So septicemia, like when something turns septic, what's the official definition? It's just in the blood?
Because I actually just had this conversation because we always call it bacteremia, which just means bacteria in the blood, right? That doesn't necessarily mean that they're causing a disease. Like when you brush your teeth, you get bacteremia because you break that barrier between your teeth and your gums. And so bacteria can get into your bloodstream, but you're fine, right? So sepsis is like a step further when the bacteria sort of spread to all your other organs and start causing like multiple organ shutdown. So with staph, it's really commonly to get kidney failure when you go septic. And that's actually what kills people.
Oh, that's terrifying.
Yeah.
But yeah, so you're PI, Dr. Conlon, right?
Yeah.
Just went on an episode of a nearby podcast, like probably across the street from us, is where at least I recorded. Curiosity, I've been on his show, Calvin's show, before. We'll link that in the show notes. And I think it's a great intro to like the mechanisms of antibiotic resistance, which is different from tolerance, you said in your 3-minute thesis, which we will also link. So if anyone wants just 3 minutes of what Jenna and her lab are working on, we'll link that as well. It's a really great rundown to get people like interested. But I didn't know there were 2 states of bacteria not responding to antibiotics, I guess, if that would be it,
that's the biggest hurdle we've faced. And I faced this in my oral exam too, just sort of explaining the difference and how important the tolerance aspect of it is. And so I think for me, it really like sedimented the difference when you think about, so when people talk about staph, they talk about MRSA, right? Methicillin resistant staph. MRSA infections take over the news. You see it all over like staph superbugs, all that. But we don't actually use methicillin or its cousin antibiotics, oxacillin, or any of these like cell wall acting antibiotics to treat staph, because there's high rates of resistance against them, right?
So they're already broken, basically. They're already obsolete. Yeah.
Right. So most commonly they use this antibiotic called vancomycin, and these people are on IV vanc for 2 to 6 weeks. So they're just getting it continuously in their bloodstream. And we don't really see resistance to that, which is why they use it. But then there's issues with like, why do you have to be on it for 6 weeks, right? Like, why isn't that antibiotic working? And like all the lab tests they do, if you go to the doctor with this infection, they'll identify if something's resistant, and no doctor is going to prescribe an antibiotic that they know the bug is resistant to, right? So it's just sort of this like weird phenomenon that seems to be really specific to the bacteria being in the human body. Because if you take them out of the human body, you won't see tolerance, right? So like if you do an antibiotic screen in a test tube, it'll look like it's sensitive, but then that antibiotic isn't working in the person. And it's just sort of this like what the heck is happening, right?
Yeah, I'm glad you mentioned that. And that's a huge problem with a lot of preclinical research avenues, right? So we think of a lot of research as people moving liquids in and out of test tubes, and for some people that's what a lot of it is. But then there's the in vivo, like inside of a living animal part of research, which can be more accurate in terms of translating it to what actually works in a human body. But you can't always do that, right?
Well, and the worst thing about staph, there's a lot of bad things, but it's a human adapted pathogen. So we tend to use mice as our model organism, and they tell us a fair amount, right? Like a lot of staph research is on facts, literally, of mice. But there are certain aspects of staph infections that you can't find in a mouse infection model, right? And we can't go around using human subjects. So there's just sort of this black box of like, is this actually happening in humans? We don't actually know, but like all the scientific evidence points to it, at least in our as close as possible systems.
So what kind of differences are you looking at between mice and humans there, or that's not really the focus?
Yeah, so our lab doesn't really study that. Victor Torres at NYU is really into that like host specificity, why staph is, or like what parts of staph are so specific to humans. And staph produces a bunch of toxins. And he's found that one of its main toxins only works when in a human host and doesn't work in mice. So like, or isn't expressed in mice, right? So if you put this strain or strain of staph into mice, this toxin won't have any effect on infection outcome. But if you put it on human cells, it's awful and completely just like obliterates the cells.
So it's found a vulnerability in our
yeah
system or something like that.
Yeah.
That kind of brings me to your research topic, which was, and correct me if I'm wrong in this, it seemed like your research is investigating why bacteria is somehow hiding out in macrophages almost.
Yeah. So macrophages are a special type of immune cell. They're part of your innate immune system. So it's your sort of first line of defense once something gets into you that's not supposed to be there. And for a long time, staph was considered an extracellular pathogen, meaning that it never got into host cells. And I don't really know why, because if you go into the literature, starting in like the 50s, we have evidence that staph can survive inside
it's
cells.
Like a flesh-eating bacteria,
right?
So.
Right. So you would think that it can get into cells. And a lot of great work from Paul Cube's group has shown that it actually, as soon as it gets into the blood, gets picked up by these macrophages. And this is sort of like twofold. So vancomycin can't get into macrophages. The class of antibiotic just has really poor penetrance. So it's like protected in that sort of indirect way, but then using an antibiotic, rifampicin, that can get into the macrophage, we found that like staph inside this macrophage is still somehow tolerant, and it's not just sort of this passive protection by like hiding in a cell that the antibiotic can't access. And so it started with Sarah, the lead author on the paper, looking into this, and then my project is sort of building on the back of that.
Okay, nice. I did catch a little bit of the paper, which dropped, what, 2-3 days ago?
Yeah,
or something
2-3 days ago.
So congrats on that. I know it's always another thing to add to your list of what you've done. And it's a huge undertaking, you know, going through it myself. So yeah, quick pat on the back, absolutely, for you and Sarah and everyone in the Conlon Lab. But it struck me really as surprising that the normal mechanisms that the human body wants to use against these things are kind of bad in the end, right? Like,
is favorite topic.
Is it a necessary? All right, so we should probably break that down. So again, macrophages, they're like your frontline host defense cell, and then you could probably explain it better. They use reactive oxygen species, right? Those are their main weapons, or
so when macrophage, so they're called phagocytes because they phagocytose things, which means they take them up into these little vacuoles inside the cell called phagosomes. And
they just eat and chew them up, basically.
Yeah, through a bunch of different mechanisms, right? So reactive oxygen species are definitely a big component of that. There's reactive nitrogen species, which are basically just like toxic forms of gases that the bacteria need to survive. They deplete nutrients in that local phagosome environment. They like really strongly acidify that environment. There's all these innate immune effectors, so antimicrobial peptides, things that are like targeted, supposed to kill these bacteria. And for the most part, they work. And the macrophages kill 90% of the staph, but there's that 10% that hangs out that like isn't killed, and that seems to be the problem.
And then they come back, right?
Right. So tolerance is sneaky in that way, and that like it kind of makes it seem like maybe the infection's gone, right? Because like it's only this small, I think of them as sort of like napping cells, right? So they're not growing, so you don't see this like take off of the infection, but they're also not being killed by the antibiotic. And so if you stop taking your antibiotic, now like they go into this state in response to stress, and so that antibiotic is a stressor, right? And so if you remove the stress, then they can wake back up. And if that macrophage happened to go to another organ, or if the staph happened to get out of the macrophage, then they spread to other parts of your body. So you might have had a small local infection that now has spread to your livers and kidneys and your spleen and your bones and your heart, like all this crazy stuff.
I'm gonna wash.
Wash your hands
after going, on the wrestling mats. Yeah, again, that's just terrifying. So the macrophages can't detect that they have staph in them, or it's just they can't get rid of it? Like, does the body still know that there's a latent infection, like in a lot of diseases? There's no broader inflammatory thing going on. Okay.
So initially, when the staph gets into the macrophages, you're going to have this really like pro-inflammatory response to it. Your macrophages are basically going to signal to all your other immune cells that something's not right, and they need backup. And so what happens is another innate immune cell called a neutrophil, which is like this really potent, badass, like killer cell comes in and just starts
SWAT
like,
team.
yeah, honestly though, and starts like deploying all of these things to try to kill whatever's in the environment.
Okay.
But because staph is able to hide in that macrophage, the macrophage doesn't now realize, it thinks it killed it, right? Because it did what it was supposed to do, put it in the phagosome, launched all this stuff at it. And so it stops sending those signals. And so not only is the staph in the phagosome protected from those neutrophils anyway, just sort of like as a physical barrier, but it's going to then cause the macrophages to stop sending those signals, so the neutrophils are going to go away.
Now, this might be something you don't know about, but I know there's some viruses that will co-opt cellular machinery and make it work for itself. And in this analogy where like the neutrophils are the SWAT team and the macrophages are maybe just the regular police force, is it possible that Staph aureus is like somehow messaging macrophages to not do anything and like stall themselves?
Yeah, so there's some interesting work on that in the context of biofilms. So biofilms are probably one of the more famous like bacterial situations, but it's basically this sticky blob of bacteria that grow on something. And there's some work showing that staph in that biofilm wants the immune system to be in a certain phase so that it doesn't kill it. So there's this sort of like pro-inflammatory and anti-inflammatory immune response. And the pro-inflammatory is really good at killing, and the anti-inflammatory is more involved in like wound healing and fixing everything once the damage has been caused. And so there's a little bit of work showing that like maybe staph somehow forces these macrophages to behave more anti-inflammatorily, that's not a word, so that they can survive in this biofilm. So there's a little bit of evidence for it. It's one of those things that's like, ooh, it's not the same way viruses do, right? They don't like, at least we don't know. Can't say it. We don't know.
I would assume, yes.
Yeah.
Which is probably dangerous in the realm of science. But for context, some viruses will actually turn on genes or have the host express their own version of a gene, kind of like a computer virus injects its own code into a computer. But anyway, so that's a tangent. So again, very fascinating. Your work is identifying that macrophages are kind of this safe haven, and perhaps unwitting safe haven, these bad cops that are sheltering criminals, maybe, if we follow our analogy, right? What do you do about that? Like, what's the next,
So that's what we're trying to figure out, right?
Yeah.
So it's this very fine line, because the macrophages are really important. Like if you get rid of all the macrophages in a mouse, they will succumb to infection really quickly, right? Like, so the macrophages are really important, and that initial pro-inflammatory response is really important, right? Like you want the macrophages to kill whatever they can. So we're kind of looking at how we can like fine tune our ability to like manipulate the immune system. So let that pro-inflammatory response happen, but then stop it at just the right point to where we prevent tolerance from happening, and then hit the staph with the antibiotics at that point before they have the chance to go into this napping state. That's what we're hoping to do, because that'd be sick. But the alternative is to also just figure out how to wake staph back up without causing like secondary infections, right? So like wake it up in sort of like a programmed way. And so that like we can intentionally wake it up and then intentionally deliver an antibiotic so it doesn't have the chance to spread to other organs, and sort of like comes to life and thinks everything's great and then immediately gets killed with the antibiotic. That's the hope. That's what I'll be doing for the next 4 years. So
I mean, it's absolutely worth the time, and it does take time, right? Because you have to grow these, and then you have to work with mice, and then let's talk about what you're actually doing in the lab, because it's really hard to visualize this stuff. I'm seeing it all within a cell, but that's not what you're looking at day to day, I would guess.
Yeah, so my project sort of has 3 separate models. So we talk about in vitro, means in glass. That's like test tube science, right? So means different things to different people. We think of in vitro as just like staph in broth, which is actually just like fancy chicken broth that staph likes to grow in.
Okay.
And we just grow it in a test tube, and we can really intentionally treat it with different compounds and different antibiotics and see how it responds in probably the most simplified system possible, because we know it's just staph. They're only interacting with each other. We also have a cell culture model. So we have these macrophages that have been immortalized. I don't know if you've ever gone into cell culture on here. Okay. Not
immortalized microglia, which are like
all right.
the cousin of macrophages.
So we have these like immortalized macrophages that we can infect. So now we have a little bit more complex system because it's staph interacting with a host cell, but we don't have all the other convoluting things like the other innate immune cells and just the host in general. And then we also have a mouse model. So we do a sepsis model. So we just inject staph straight into the bloodstream of mice. They don't look or act sick. So that is very comforting to me that they probably don't feel too well, but it's not one of those nasty infections where they like actively look sick, which helps me do the most work. But
it's hard enough as it is.
Yeah. Yeah, it would be really sad if they like looked upset about it, but they seem okay for the most part.
Nice. So you have this combination, and then ultimately the hope would be move it clinical. Now I know we're very, very far away from that, right?
Yeah.
But hopefully, like the paper that your lab just published will at least let doctors know that this could be part of the problem, right? Are there any people in hospitals like checking macrophages now for staph to see? Because I mean, you can isolate macrophages. A lab next door to mine does this.
Yeah.
They get blood shipped in from human patients. They isolate the macrophages, and then they can like study those for experiments.
Yeah.
I think mostly in HIV context. But couldn't you also check if there was staph inside of those?
Yeah, so ultimately that'd be great, right? I don't know how often that's actually done in the clinic. I do know I just saw a case study came out of Vietnam where they took patients that had come to a hospital with staph infections and found that a lot of them had this sort of tolerant phenotype, and I assumed that it had been intracellular. They also like then took the staph and infected cells and saw that it was tolerant to the antibiotic. So it seems that like people are sort of catching on to this idea. It's been shown for other pathogens as well, so it's not something that's unique to staph. I think how it gets implemented in the clinic will be interesting to see. There's a lot of focus on just making some of these, like vancomycin, that antibiotic that can't get into the cells, just making it so it can access the cell, sort of taking the antibiotics we already have and making them more effective. So I don't know how those two things will sort of come together clinically. But I think it'd be really interesting to see how that plays out with patients. I think it's important. So.
Yeah, we'll keep an eye on it. Absolutely. So in the lab, you're working in tissue culture hood, which I know what it looks like, but for other people, it's trying to like think of an analogy.
Big silver box. I don't know.
Big shiny box, and you only put your arms inside of it, and you're keeping everything.
Oh, like babies, like in the I don't know.
Oh yeah.
I don't know if that helps at all in the newborn ward. If there's like a premature baby, they have them in those little boxes with just like the gloves.
Yeah, exactly. So
Kinda
only
like
your arms go
that.
in, and then everything is sterilized and very contained. Then when you work at the mice, I'm assuming you're looking like at their macrophages, you're looking at their spleen, the immune, like a big immune cell hub. So day to day, I just try and like help people visualize like what you're looking at on any given day. What were you doing today, for example?
What was I? Oh, I actually did mouse work today, so perfect. So UNC has a bunch of different animal facilities. So they're basically these big rooms full of cages of mice that are looked over by vets and vet techs. And so I guess, so our infections usually span about 4 days, 3 to 4 days, depending on what we're doing. So we have our mice in a biosafety level 2 facility. So staph is a BSL-2 pathogen because it can infect people. So that just means there's extra safety precautions you take when working with it. So we have our mice in there, and then I work in a hood in there as well, which gets hard with the mice because you have to actually like handle them, and you have pretty limited mobility in a hood. But so they get infected in their tail vein. And then today I treated them with antibiotics. So we do what's called an intraperitoneal injection of that. So you basically, you know how like cats and dogs have the scruff on the back of their neck, you scruff the mouse by the same way, hold their tail because they'll whip it around like. And then you stick it, they have like 2 little nipples on the bottom, and you stick the needle in between there and just kind of put the antibiotic so then it spreads to the rest of their body. And then I watch them to make sure they don't have any weird adverse reaction to anything. And then ultimately I will use their spleens, livers, and kidneys and sort of just see how many bacteria are in those organs. Since we infect in the blood, it has to travel to those organs. So whether or not it gets there is really important. And then whether or not the antibiotic worked in that organ is what we're really looking at for the tolerance. We're working on figuring out a way to do like some imaging stuff to see if they're actually localized with macrophages inside the mouse, because we haven't done that. Paul Cube's group, who I mentioned earlier, does some more of that intravital imaging. So while the mice are alive, he looks at where the staph is going and what cell types it's associated with,
imaging is
which is so cool. But we don't actually look at that yet.
Yeah, I know it's really technically difficult. Just kind of on the cutting edge, I think, as a scientific community.
Yeah. Yeah.
A lot of people are looking into doing it, but it's really like fuzzy, but that's how things were 20 years ago in just regular fixed.
It's insane the things they figured out with like a fraction of the technology we have now. They still made some like life-changing discoveries. So I think it's crazy.
Yeah, no, the mouse work is definitely the hardest thing that I do day to day, and it's kind of nice to work with mice, but at the same time, you know, ultimately they are sacrificed in the end, and so that's really sad.
Yeah,
it's the hardest thing to deal with, at least for me.
My mom isn't. Yeah, that part bothers me a lot. And sometimes people will be like, "Oh, you'll get used to it," and it's kind of like I don't want to get used to it. Like I don't ever want to be okay with the fact that I am having to do this to an animal, right? Like I think it's very necessary, and I think it's very important to advance the research, but I never want it to just be something that's normal. You know, because it's not like it's still like I still don't like it, and like my parents don't like it. This part is like my mom's so upset when I talk about mouse work, which
I mean, but you have to keep the things in sight, like the 20,000 people that are dying every year from this, and if we can cut into that significantly by doing, or even marginally by doing this, you're saving many lives, and like those are very painful lives.
Yeah.
If you like what you're saying about Staph aureus is true, and like it doesn't seem to affect mice in this, really fascinating.
Yeah. It also, I guess, is comforting to me. I don't know how this like makes sense in my head, but that they were like bred to be lab mice, right? Like they were. It's not like we go into a field and pick up these happy mice that were living their wonderful life in the grass. You know, like this is sort of the life that they know, and like what their purpose was, right? So for some reason, that's comforting to me.
And honestly, when you look at the mouse rack, I can't help but think of apartment buildings.
Yeah.
And like that's how we choose to live.
Yeah, right?
So we shove ourselves into these basically mouse racks, and we just hang out with our family, which is what
they have all the food they want in the entire world, and they have like little huts and like little toys.
They don't have to worry about owls or
right. I think it would be worse to get eaten by a snake, but I guess I don't know.
I can't speak for experience on that one. But anyways, that's it's really incredible work, and makes me think again about like going to the gym and stuff like that. So one thing I always like to ask is, how has your research, or other people's even, like changed the way you think about life or act, if at all? I mean, sometimes.
Yeah, it's weird because like you would think as microbiologists we'd be a lot more conscious of that sort of stuff, but like I don't know, like if I drop something on the floor, I'm still gonna eat it. Like I don't care, you know. Like it's one of those things.
Totally fine.
Yeah, right. That's what I say. I am more aware of things like washing my hands, or like if I'm in the lab, like if I am unpacking a box and I use the scissors that are on my desk, I'm like, oh, those probably have staph all over them, so I should go make sure that I wash that off. You know, I think you're conscious about certain things. It's also just made me, I guess, more observant. Like when I see people doing things, I'm like, oh no, you have a cut on your hand, like don't touch that, which is probably super obnoxious, but
could have just saved their life.
Yeah, right. Maybe I saved them from getting a septic staph infection. You're welcome.
No, I mean, it's absolutely something to keep in consideration. And on the broader topic of like antibiotic resistance and tolerance, this is something that, again, we shouted out Calvin's podcast. Doctors have previously been prescribing antibiotics like they're candy. Like you go in, you know, I mentioned the infection I had. I knew it was fungal because someone else had already diagnosed it from the same tournament we went to. And then I went in and was like, hey, I have this fungal infection. And they're like, here's an antibiotic. I'm like, that's not how any of this works. So these are trained doctors. And they're like, just take the antibiotic. I'm like, no.
Right.
I know what this is.
I think that's one of the hardest things is like a lot of people go to the doctor when they're sick, and there's this connection in everybody's head that antibiotics means I'll feel better, right? So they could have a fungal infection or a viral infection.
Oh, it works, but yeah.
Or it's placebo. But like they'll have a viral infection or a fungal infection that antibiotics don't work against, but like that's what they want when they go to the doctor, you know? So like, can you fault the doctor? Like not really. Like their goal is to help the patient, and if that's what helps the patient, then fine. But that definitely is like misuse and overuse of antibiotics is definitely like the major contributor to resistance.
And then also, if you can prevent yourself from getting infected in the first place, then you don't need to even ask about an antibiotic or be told to take one. So stay healthy. You know, although I preach way too much, probably eat right, get your sleep and stuff like that. But being a grad student, that's not always an option. You know, free time is hard to come by. When you do get a chance, what's your free time? What's your guilty pleasure outside of the lab?
Guilty pleasure, reality TV for sure. I get made fun of a lot for that, but
that's probably pretty rare on campus. So, you know, don't hear that get brought up a whole lot.
I do enjoy some good reality TV.
Any particular shows? I haven't.
Oh God. So lately I'm really into 90 Day Fiance. So do you know what that is?
Nope,
Probably not. All
guess,
right.
I can guess.
It's one of my very niche things, right? Yeah. So there's this visa program called the K-1 visa. And it's basically if a person in the United States wants to marry somebody not from the United States, they can apply for this visa. And if it gets approved, from date of approval or date of receipt of the visa, I'm not sure, they have 6 months to move to the States. And then once they're in the States, they have 3 months to get married. And so it basically just follows the lives of these people that have applied for this visa. And it's like, for a lot of these people, they've only spent like a week together in real life, like in person, not in real life. And so they like basically are using these 90 days to decide if this is the person they want to marry. But if they don't, then the person has to go back to their home country, which like if that's what's best for everybody. Yeah, it's just really, it's so different from how I normally think of relationships and marriage panning out that it's really interesting to see how these people like interact and how their families handle it.
You probably get to learn about different cultures and stuff like that too, like passively through that because people are coming.
Yeah, that's been one of the more interesting things is seeing like there was a guy from Tunisia who just like they're just a much more conservative culture. And so he was just like he was uncomfortable with like PDA and stuff like that. And his fiance had a really hard time with that because that's pretty common in the States, right? To see people like holding hands and kissing in public. And then this guy was like, I can't do that. Cannot do that. And she just like could not get that through her head. And he was like, it's my culture. Like, I'm sorry, I don't. And so it's been interesting to learn about those countries that I haven't really like looked into or studied in any facet. Kind
of forgot existed after you took geography.
Yeah.
Yeah, it's hard to keep up. Absolutely. So nice. So reality TV. There's what else are like in the lab? How do you like listen to music or
lately?
Early on staring at Staph.
Yeah, just stare at Staph all the time. I actually listen to a lot of podcasts. I have this problem where if I'm listening to music that I know the words to, or if I'm listening to a podcast that I'm like really interested in, I'll get distracted from what I'm doing at the bench. So if I'm doing something that requires my attention at the bench, like a new assay or something I've never done before, it's like headphones off 100% because I'll just completely focus. But like if I'm just doing something sort of routine, like inoculating cultures or weighing out tubes for an animal experiment or something, it's headphones on, music or podcasts. It's also my signal that like I don't really want to talk.
Yeah, I mean, so if I'm like loading a gel, which is like you need precise motor control and you need to be paying attention, like classical or instrumental music only.
Yeah,
like
I do a lot of I recently started doing opera because there's still like it's classical, but there's words, but I don't understand them because they're in Italian. So
yeah, I can't distract you. Right?
Yeah, right. I can't understand it. So
until you watched a bunch of 90 Day Fiance and you get to absorb the language. Very cool. Very cool. So yeah, once again, thanks for coming on. But we always like to kind of bring in like who got into science and how. So like your path to science, what brought you here? Were you always fascinated with MRSA?
No, so I feel like I'm one of those, a bunch of my friends recently posted a bunch of like Throwback Thursday pictures of them with like their first science poster at like the middle school science fair or something. And I was never really one of those people. I was definitely a lot more math inclined growing up. It was definitely always STEM, like history and English were not my thing. But I had sort of a contrived path, I guess. So in high school, I took AP chemistry and I had the best teacher in the world, Mr. Blankenship. He was absolutely wonderful. And so I applied to college thinking that I wanted to do something chemistry adjacent. And I had this weird like determination to be a woman in engineering, even though I hate physics and I hate calculus, which is a lot of engineering. Yep. So I applied to the University of Texas as a chemical engineering major. And Texas has this program where if you graduate in a certain percentage of your high school class, you get automatic admission to any public school in Texas. It's not automatic admission into your major, I learned, but it is automatic admission into the school. So I didn't get into chemical engineering, which sucked at the time, but was definitely for the best. So I switched to chemistry, and then I had this awful chemistry professor who I will not name because he was so terrible. But basically he convinced me to like not do chemistry, which is a terrible thing. But, and so I switched to biochemistry because I was like, maybe I just need a little bit more of the biology. And then I almost failed the first semester of biochemistry. So I was like, probably not for me. And I remember meeting with my genetics professor and just being like, what the hell do I do? Like I've changed my major 3 times in 2 years. I was pre-med, pre-pharm, pre-teaching, like all this crazy stuff. And she was like, take a microbiology class and see how you like it. Like, that was the class that I really enjoyed. So I signed up for a microbiology class. And I like absolutely fell in love. Like, it was just the intro class, you know, like one of those 400-person classes. But Marvin Whiteley was my professor. He was at UT now. He's at University of Georgia. Sorry. But he was just like great. You know, he was so interested in it. So he's one of those people that because he liked it so much, you liked it. Just sort of diffusion. I don't know. So I switched my major to microbiology, and then I had another sort of like, what am I doing with my life? Like, a BS in microbiology doesn't get you very far. And I've always wanted to be a teacher, so I thought about going into teaching, but I also like school is the only thing I'm really good at. So I met with another professor, Brian Davies, who's also wonderful. And he sort of talked to me about grad school and his path to grad school and why he thought I would be a good fit. And I was like, okay, like I'll apply. Like if I get in, I'll go, you know. I was interested in it, and I liked the idea of having a research career, being like a staff scientist or something. But I figured like if I didn't get in, then that would be my signal that it wasn't for me, and I would figure it out from there. So I'm here because of Brian Davies, but he convinced me to apply, and like I love it. I absolutely love it. And working in a lab in undergrad like completely changed my perspective on science and the research community and everything. And I think that was really critical to me ending up here. I love. So.
Yeah, I don't think that is like that rare of a path. Like, it's very similar to mine in that I had several unfortunate years of science classes. Let's just put it that way, in my formative years. And I had all but written it off. And then finally I was like, hey, I actually really like this stuff when you have a competent or exciting professor.
Right. It sucks that like one bad professor, like my chemistry professor, like I thought about dropping out of college. Like he told me I didn't belong in college and that I didn't belong in science. And like that was sort of how I identified, right? It was like somebody that was good at school. And so like that really sucked. And I'm really glad that I didn't let that stop me. Like, I don't know what would have happened if I dropped out of college. Like that would have been insane. So I'm very thankful for the couple professors that were, you know, engaged with like my path and were really excited for me to get into science. I think as scientists, we should, like, if you like your job, why wouldn't you want other people to join the same field, right? Like, you're like, this is sick, you should come do it. But for some reason, this guy was like, no, you don't belong here. And I was just like,
oh, yeah, some people just have a bad day every day.
Yeah.
I can't explain it. So yeah, any advice for people? Probably like stick it through would be a common theme here.
Yeah.
But anything else people, maybe microbiology specifically, I don't know.
Microbiology
There's
specifically.
anything that like helps on that track?
I think one of the biggest pieces of advice, so Michael Johnson was just here. He's a professor or assistant professor at University of Arizona. And he was just here, and he said that his biggest thing was fortune favors the bold. I think that's what it is. And like, don't tell yourself no, let other people tell you no, which sounds bad, but it's true, right? Like, why not just apply? Because the answer might be yes. He's actually the reason I did the 3-minute thesis contest, and that worked out wonderfully for me. Yeah. So,
oh yeah, by the way, Jenna got People's Choice Award. Yeah. I didn't say that. That's how I found out about her.
I was People's Choice winner for 3-minute thesis. That was exciting. So I think it is important to just like shoot your shot, right? Like go for it. You never know what could happen if you do it. And I think that's really important in all facets of life. But another one is to separate your professional success from your self-worth. Especially in science, we fail a lot, right? Like that's just what happens. Experiments don't work. Grants don't get funded. Shit sucks. But that doesn't mean that you're not a good person and that you don't have other great qualities. And I think that's been really important as I move along in grad school, is not thinking that like one bad experiment day means that I shouldn't be a scientist and that I have bad hands and I don't belong here, you know. I think those 2/3.
No, absolutely. If it was easy, like we would have cured everything.
Right.
Right. There's obviously a lot of interest in these things working out, but you throw the best people at it and sometimes it still doesn't.
Yeah.
Thank you, Jenna. It's been great.
Yeah, thank you.
Any last thoughts before we wrap it up?
Ooh, not on the spot.
All right,
I'll probably think of 1,000 things later, but
yeah, absolutely. And then I will of course ask you for some of those links in the show notes. People can y'all can refer there. But otherwise, yeah. Thanks for coming in.
Yeah, thank you.
Thanks for listening all.